Hello and welcome to this Bio43 pre-lab video. This is a one-off lab on bioinformatics, but it links back to our first unit, where we studied Tetrahymena as a model organism. Tetrahymena makes a good model organism for cilia function because of the similarities in cilia structure. Now we're going to look for similarities on the genetic and protein levels. In doing so, you'll learn about the computational tools commonly used in molecular biology labs. So the first step is picking a gene you want to know more about. To do that, let's visit the National Center for Biotechnology Information, or NCBI's, gene database. If the site design looks a little familiar, it's because this is the same organization that runs the PubMed database of journal articles. If you already know the gene you want to study, you can search for it directly. For example, if I wanted to know more about human beta-galactosidase or lactase, I can type in its gene name, LCT, choose the Homo sapiens version, and the gene comes right up. But we've already studied this enzyme, so let's pick something new. I'll go to Advanced Search so I can specify the organism. Let's stick with a human gene for this example. I like to study the innate immune system, so I'll search for that. But your focus will be on genes involved in ciliopathy diseases or conditions. This gives me a fair number of hits. I could narrow it down with more specific search terms, or scan the list for things that interest me. But for now, let's just look at the first hit. There is a lot of information here, but I'm just going to cover a few key parts. The summary provides a good overview. This gene has the official symbol MEFV, and is known as the MEFV innate immune regulator, or PIRIN. And it has these alternative names. This section also contains a brief description of the gene and where it is expressed. There are more expression details a little further down. We see the gene is expressed at low levels in a bunch of different tissues, but is highest in the appendix, bone marrow, and spleen, which makes sense for a gene connected to innate immunity. Next you have the bibliography, a list of PubMed articles that reference this gene. This is an excellent place to start learning more in-depth details about your gene of interest. Sections like pathways, interactions, and gene ontology can tell you more about the gene's role. The final things we want here are the sequences of the gene and the protein produced by it. We'll skip over the genomic sequence because this includes introns that we don't need to worry about. So let's jump to mRNA and proteins. This gene actually makes a couple different gene products, or isoforms. If this is the case for your gene, you can look at past research to decide which one to go ahead with. Let's just choose isoform 1 here. The first link will take you to the mRNA sequence, while the second leads to the amino acid sequence. Record both of their NCBI reference numbers. Clicking the links actually gives you a lot more than just the mRNA and amino acid sequences. But for now, if you just click on FASTA, you can get those sequences alone. We can learn more about the gene from this sequence info. First, let's find genes with similar sequences using another NCBI tool, BLAST, short for Basic Local Alignment Search Tool. The NCBI database has the sequences of a huge number of genes from many different species. BLAST will take a sequence you're interested in, the query, and find genes with similar sequences, or homologs. High similarity implies a shared evolutionary history, that the two sequences can be traced back to a common ancestral gene. So if you don't know the function of your gene, but you find that its sequence is similar to another gene with a known function, it would be reasonable to hypothesize that your unknown gene has a similar function to that of the homolog. You can search for homologs at both the nucleotide and amino acid level. Let's look at a nucleotide BLAST. Enter the mRNA as a query sequence. You could copy and paste the full sequence here, or just the NCBI reference number. We won't change any default settings, though you could choose to limit your search to certain organisms or taxa if you wanted to. Also, we'll stick to highly similar sequences for now, but if you don't get any results in your initial search, you can loosen the criteria and allow somewhat similar or more dissimilar sequences. Now we'll click BLAST to start the process. This can take a little while if many people are using BLAST at the same time, but will generally be less than a minute. The table at the bottom contains a list of genes with similar sequences to our query. The top hit is the mRNA sequence we input, which makes sense. Let's exclude humans from the search to see what homologs we get from related species. Unsurprisingly, we see closely related sequences from our closest relatives: chimpanzees, bonobos, gorillas. We can exclude all primates to see if there are homologs for more distantly related species. Ooh, chinchillas. Looks like it's predicted to have the same function as its human homolog. There are several important numbers you can find here. Query cover tells you what percentage of the query sequence the program was able to align with this match. 51% in this case. The e-value tells you the chance of finding this strong a match by random chance given the size of the database. Here, it's so low it rounds to 0, so it is extremely likely that this match is due to shared evolutionary history. Then percent identity tells you the percent of nucleotides in the aligned region that are identical. Click on the match to see the alignment. The top line is the query sequence we entered, and the bottom is the chinchilla gene it matched with. The vertical lines indicate identities, bases that are the same. Sometimes you'll see dashes. That's where gaps needed to be inserted in one sequence or the other to make the alignment work. So this gap indicates that sometime after human and chinchilla lineages diverged, either 3 extra nucleotides were inserted in the human gene, or 3 nucleotides were deleted in the chinchilla gene. Information like this can tell you much about the evolutionary history of the gene. While NCBI BLAST is a great broad search engine, if you're searching for homologs in a particular species, several model organisms have their own databases that can be more detailed and up-to-date. For example, I study solanaceous plants and use the Sol Genomics network. You'll use the Tetrahymena Genome Database Wiki to search for Tetrahymena homologs. And you'll do your BLAST search at the protein level. But the same principles apply, and the e-value will still be the key criterion for assessing the quality of matches. Now let's look at other ways to learn more about our protein of interest. The first protein resource we'll use is ExPaC, the Expert Protein Analysis System. Their ProParam tool gives us a simple breakdown of our protein based on the amino acids present. ExPaC doesn't recognize the NCBI reference number, so we'll paste in the full amino acid sequence. And compute parameters. This gives us information like the number of amino acids in the protein, its molecular weight, and the amino acid breakdown, highlighting the number of positively and negatively charged amino acids. The final tool we will look at today is called PFAM. This is another program that examines the protein's amino acid sequence. It searches for common domains, portions of the protein with known functions. So if we don't know a protein's role, finding what domains it has can help us narrow things down. Let's search with our sequence. And we see 4 significant matches. The cartoon at the top tells you where along the protein the domain matches can be found. And the table below has more details. You can click on the family name to learn more about a domain. Let's find out more about these B-box zinc finger domains. From this summary, I now know that this domain is found in a variety of proteins, including transcription factors, ribonucleoproteins, and proto-oncoproteins. And proteins with this domain are often involved in ubiquitinylation. So if nothing else were known about this protein, we could start by investigating those possibilities. Those are the bioinformatics tools we will be using in lab. And that does it for pre-lab videos. So long!